Yes, hello. Thank you for the introduction and thank you for having me at this amazing talk. My name is Jason. I'm a PhD student from the Biological Engineering department at MIT. And in the past few years, we developed this academic solution to single-cell BCR sequencing, and we called it B-Seq. And we've been using it to study various vaccine responses. And the main benefit of it is you can recover full-length BCR sequencing from the 3' barcoded single-cell RNA sequencing libraries. So, as a quick recap for those who don't use single-cell RNA sequencing, it's a pretty powerful tool to study a lot of heterogeneity in tissues at a much higher analytical resolution that you can't really do with like flow cytometry. It has four general steps. So after getting the single cell suspensions, we would compartmentalize individual cells with a capture bead uh, that are DNA barcoded. And then the cells are lysed and their mRNAs are captured onto the capture beads. And after many rounds of PCR, you can make a library for sequencing. And to this day, still most people use Illumina sequencing, which is a version of the short read sequencing. So now if we zoom in onto the mRNA capture step, the most widely used method and the more traditional method, it's still using a poly-DT capture sequence that captures the polyadenylated ends of the mRNA. And as a result, the cell barcode that is on the capture bead gets conjugated to the 3' end of the mRNA or the cDNA after reverse transcription. And that's why this is called the 3' barcoding protocol or chemistry. However, the 3' barcoding has some limitations when it comes to study adaptive immune response, such as T and B cells. So let's say this cDNA is a BCR transcript. Um, because the variable regions are on the 5' end, when you try to make the libraries, you inevitably have to fragment it the cDNAs to fit on Illumina sequencers. And because of that, you lose the variable region, which is the most important part of BCR or TCR that specifies what they target. So to overcome this, we developed a method called B-Seq that can simultaneously profile the BCR sequences from the 3' barcoded gene expression sequencing. The way it works is that after generating the cDNAs, we would split the cDNA library, or for people who do single RNA sequencing, the products are called the WTA, whole transcriptome amplification products. We would use half of them to generate the gene expression library and use the other half to generate the BCR sequencing library. And the way we do that is we would design custom primes that are probes, sorry, not primes, primers, but also probes that are polyadenylated that can bind to the constant regions of the BCR transcripts. And because they're polyadenylated, because they're they're biatenylated, we can use streptavidin to pull them out and enrich for only the BCRs. And after the enrichment steps and further refinement, we can read out the full-length variable region with two overlapping reads using a set of custom primers that bind to the constant region, and the other half are just standard Illumina primers. And then after reading them out, we've been using Immcantation for the processing of them to build the final contig. And here are two use case examples that B-Seq is applicable to a wide range of 3' single-cell RNA sequencing protocols. On the left, for both of these experiments, we just took human PBMC and did single-cell RNA-seq on them. On the left, we used SeqWell S3 protocol, which was a method that was developed at MIT by our lab and our collaborator lab, Alex Shalake's lab. On the right is this is the commercial solution that most people use, 10x, but the 3' version of the 10x genomics kit. To demonstrate that we can recover sequences that are highly specific, we did this experiment where we transfected hex cells with a known BCR sequence, both heavy and light chain in separate plasmids. And then we mixed the hex cells with human PBMC and performed single-cell RNA sequencing and B-seq on them. And as you can see by the UMAP, we identified different versions of the B cells, naive memory plasmablasts, and then a cluster of hex cells, and then various other cell types. And among the among the hex cells, you can see that we recovered this sequence from almost over 98%. Over 98% of the U6 BCR sequences were recovered from hex cells and not from any other cell type. So it really shows that it can recover BCR sequences from their respective cells. And on the other hand, if we look at how many mutations that were accumulated on these sequences, because hex cells don't go through somatic hypermutation, so this is just pure protocol errors. We see that about 90% of the sequences have no errors, and about 95% of them have one or less number of bases that were erroneous. So another thing that we wanted to check was how does it compare? I'm sure a lot of you might have thought of this already also. How does it compare to the 10x genomics 5' kid? I mean, this is a big problem in the study of T and B cells. So as the leader in the space, 10x kind of made their versions of the solution, which is to move the barcode on the 5' end. And in this case, the way they do it is they also split the cDNA libraries into two parts. They use half of it to do a whole transcriptome sequencing, and the other half they do two nested PCRs to purify out the BCR sequences. And then they built them in silico to construct the full contig. So to compare with the 10x 5' kid, we went into the literature and spotted a few studies in the context of vaccines where they used the mice, human and human tonsil. I think human tonsil is the middle one. This study used human lymph node. And we kind of the way we did this analysis, it's kind of asking if we bin the B cells into these bins on how many BCR reads that we recovered from the whole transcriptome. So if we, for example, in this here we identified B cells that have 5 transcripts that were recovered from the gene expression, what is the probability of these methods that can recover a BCR sequence? So on the left, these are heavy chains. On the right, these are light chains. So the number one trend I wish to point you to is the fact that when you have more BCR transcripts in the cell, you are more likely to recover a BCR sequence. So that's kind of standard and not too super surprising. Now, if you look at the blue color tones, which are 10x 5' and the red color tones are using 3' single-cell RNA sequencing protocol, but applied with B-seq, our method can recover the BCRs with similar probability, if not better, which is very exciting to us. So to actually apply this, we have a lot more study in this space, but this is just a proof of concept to show that our method is high fidelity. So in this case, we immunized rhesus macaques with a pneumococcal vaccine, serotype 3. It's a carbohydrate conjugated to a protein. And then on week 4, we isolate out their PBMCs, their blood, and we sorted out the antigen binding B cells. As you can see, here is another portion where we use the Immcantation's pipeline, which is super fabulous. On the left, these are unvaccinated adult monkeys. On the right, these are vaccinated infant monkeys. And the color bar shows the clonal expansion of the cells. So without immunization, these monkeys don't really demonstrate antigen specificity. Even if they do, these cells aren't clonally expanded, meaning that they're not truly activated. On the right, you can see this one monkey developed a very strong clonal expansion, suggesting a stronger immune response to the vaccine. So we picked 20 antibodies or 20 antibodies from the top 20 clones and expressed them based on what the B3E-Seq was able to recover and actually ran Luminex to test their binding specificity. Among the 20 of them, the 3 kind of showed like heterogeneity in their specificity, but the majority of them showed high specificity, suggesting that not only it's functional, it was able to recapitulate the B cell response. So in conclusion, B-Seq B-Seq can simultaneously resolve single-cell transcriptomics and also recover high fidelity and full-length heavy and light chain paired BCR repertoire, which it was traditionally not able to be accomplished with the 3' protocol using traditional Illumina sequencer. And this work was not possible without my lab mates, especially Duncan Morgan, who was a senior graduate student when I first joined the lab. He started out this project, and I kind of pushed it to the finish line. And with the two of us together, we put the methods together. Hopefully, it's useful. This is an academic solution to a problem that kind of bothered a lot of people in the field.
Thank you, Jason. Let's see if some question pops in the chat.
I think Uri had a question. Maybe you can unmute.
Yeah, can I just ask it in audio because I'm having a hard time typing?
Yep.
I'm just wondering, could you tell us, like, compared to 10x, how many cells do you get, like, per sample, or how deep is the sequencing in terms of repertoire size that you get compared to 10x?
Yeah, thanks for the question. We in the paper we have a benchmarking on like recommendations on how deep you should sequence. The method itself isn't that different to the recommended sequencing depth as 10x suggested. So you kind of back calculate, for example, if you want 100 read per cell, and you know how many cells you have per sample based on your transcriptome data, and then you just kind of back calculate on how deep you want your or how to choose your sequencer.
So
If
typically,
that
you
was
would
your
get,
question.
so typically you would get the same amount of cells as you would get in a 10x experiment in the end, like when. So
Oh,
usually, or could you get more because you're only
yeah,
looking for B cell and T cell receptors?
yeah. So I think the question you were asking has 2 parts. One of them is like the recovery of cells based on what the initial protocol you use. So with both 10x and SQL or 10x 3', the recovery, that's stated by their protocol. But if you were asking what fraction of the cells you were able to recover a BCR sequence from, this is the analysis that we did. So the conclusion was you can recover about the same proportions of BCRs using B-Seq or the 5' kit.
Okay, thank you.
Thank you, Jason. Maybe you can drop as well the link to the
I think
publication
there's
in
one
the
more
chat.
question in the chat.
Oh yeah. This is also what's the benefit of choosing B-Seq over 10x 5 prime? I wouldn't say there is a huge well, the benefit is cost. It's cheaper. And also more flexible. So if you have archived samples using their 3 prime kit before they had 5 prime kit, you can now apply B-Seq to recover BCR or TCR information from something that was just impossible before. Another benefit is if you're choosing 10x or some more academic solution like DropSeq or SQL or other just academic solutions, it would be cheaper to do these kind of experiment than 10x because the most of the cost comes from their gem beads. But both of them work well. So we're not trying to say we're better than them, but it's just an alternative.
All right, thanks again.
Thank you.